Good morning, everyone. My name is Anna-Liisa Fabritius, and I'm the Operations Director for the Humboldt County Growers Alliance. HCGA is the trade association representing licensed cannabis businesses in Humboldt County. We are pleased to present to you a week of education with our Cannabis Genetics and Bioethics Conference that is produced in partnership with Hendricks Farms. Thank you to Daniel Hendricks here. Every day this week at 11 a.m., we'll be bringing to you different tools of the trade. Yesterday, we hosted Chris Verney and Daniel Stein talking about microfarming. Today, we are talking about viruses, viroids, and pathology. On Wednesday, we're talking with tissue culture and meristematic restoration in cannabis. On Thursday, genetic editing for yield and stress tolerance. And on Friday at 11 a.m., genetic storage for common access. We're gonna wrap up our conference at 1 o'clock on Friday with our Cannabis Breeders' Rights and Bioethics panel featuring Nat Pennington of Humboldt Seed Company, Jerry Whiting, LeBlanc CNE, Jesse Dodd, BioVortex, Sunshine Johnson with Sunbolt Grown, Eleanor Kunst of LeafWorks DNA, and this panel is going to be moderated by Colleen Byers from Breeders' Best. It's during that panel that we're going to be discussing everything that's been presented over the week. All of these presentations need to be registered for independently, so if you've not registered for the following panels, make sure you do so at hcga.co/Cannabis Bioethics and Genetics Conference. And now I would like to introduce my colleague, Heather Luther, to talk about how we are being able to bring these presentations to you.
Hi, everyone. So we're really happy to be able to provide these presentations for free, and that's thanks to our sponsors. So our sponsors for all week long are Tag Risk Insurance, Rocky Mountain Cannabis Consulting, RadSource Technology, Volks Amendments, and the HCBDC, which is the Humboldt County Business Development Center, in conjunction with a grant from Headwaters Fund. So our main sponsor today is Tag Risk Insurance. Did you know that Tag Risk Insurance just saved Emerald Sun over $60,000 a year on their workers' comp, and you could be next? At Tag Risk, they specialize in insuring the hospitality and cannabis industries, which enables them to negotiate the best rates and coverage for their customers. On average, they save new clients 20 to 40% off their current insurance costs and help eliminate coverage gaps. Some notable companies they insure are Emerald Sun, Amaranth Farms, and the Microbuttery. Tag Risk offers commercial property, general liability, workers' compensation, crop product liability, cargo, EPLI, and more. So if you're interested in saving on your current insurance and upgrading your coverage, call or email today. Call Larry Anaya, and his cell phone is 714-553-9171. Or you can just visit tagrisk.com, license number OB67660. And I'm going to post that info in the chat screen on the side as well. I definitely recommend reaching out to Larry, even if you just want to chat. He's a great guy. So again, thanks to our sponsors.
Thank you, Heather. And with that, I'd like to kick it over to Daniel Hendricks of Hendricks Farms to introduce our panel and get us going.
Hey everyone, welcome. We put this whole presentation together based on just some really amazing speakers and content that we were exposed to over the last year and a half, 2 years. And we wanted to make it accessible to everyone. And so this, it was really, came from Joanna, Joanna Berg's Dirty Business Soil. She just does wonderful work and has helped us really advance our nursery, and we think that everybody would really benefit from this information. So enjoy it. Thanks, Joanna. You want to take it from here?
Yeah. Thank you. Thank you, Daniel and Hendricks and HCGA for
Hey, Joanna, I don't know if you're
on this conference.
Joanna,
really
am
excited to be out here today, virtually out here, and sharing with
Hey, Joanna. Joanna, your sound is not coming through. Doop, doop, doop. Joanna. Oh no, what is going on?
Here, we'll hold on, everybody. We just have a little connectivity issue. So I hopefully can get a hold of Joanna directly, and we'll pivot in just 1 minute here. Okay, so I messaged with Joanna, and it looks like the Zoom it wasn't able to link up, and so let's see. Do you want to test it one more time, Joanna? Test your audio.
Yeah, can you hear me okay?
Yeah.
Yeah.
Is that good?
Yeah.
Okay, my internet connection is something's going on, but of course it's going to happen the second that I need to start talking on the webinar. So charming.
Joanna, if we start to lose you again, it might be better to turn off your video and just that way you'll still be participating, but it will
focus my bandwidth on audio.
Yeah.
Got it.
Okay.
Great.
Okay.
So now you have to start again because we couldn't hear you. We're a bit of a robot.
Okay. Well, thanks for everyone being here from Humboldt and beyond. I know we have people from far and away tuning in virtually, and it's a pleasure to be here. Thanks to HCGA for putting on this bioethics conference in conjunction with Hendrix. You guys are awesome. And, you know, Dirty Business has been working with AL&L Crop Solutions since 2014, Anna-Liisa. I think we decided it was 2014. So we've been having a relationship around plant disease for several years. So I'm really excited to bring Anna-Liisa Fabritius from AL&L Crop Solutions. She has a Ph.D. in plant pathology from UC Riverside, and she's been working in the plant pathology diagnostic industry for many, many years. I want to hand over the screen to Anna-Liisa so that she can share some information with us. And feel free to message me questions either on the instant message feature, the chat feature, or the Q&A feature, and we'll try to get some questions answered as we go along. Anna-Liisa?
Thank you, Joanna. I am here. I'm going to be sharing my host has disabled attendees screen sharing. So Daniel, can you let me share my screen, please?
Yep, making, let me make that happen right this minute. There you go. Okay.
All right, my share, there it is, share. And let's put that over there. So thank you, Danielle, for letting me come in here and talk to your audience today. Thank you, Joanna, for making this happening for me. Let me put you up on there in the panel so I can see you. So like my title shows it, I was invited to talk on the viruses and viroids and other disease of cannabis and what should every grower know. So feel free to, if you have any questions coming during the talk, after any slides, Joanna will be moderating this talk. So I can answer questions, Joanna can answer questions. So whatever, feel free to ask your questions. So I am a plant pathologist at this company called AL&L Crop Solutions. We are located in California, Northern California, in Vacaville. We have been in business since 2009 and working with cannabis diseases probably since 2012. And some point of this presentation may be coming really technology heavy, but I felt that it's important for you to know some of the techniques, the basics of the techniques that we are using for plant pathology disease diagnosis. So you may just let it go, or if you want to get further into the asking questions, that's fine with me too. So today's presentation will start about just in general about plant diseases, diagnosis and detection. That's what we do in this laboratory of ours. Plants like us can get sick. There are lots of different kind of disease-causing agents, viruses, bacteria, fungi, oomycetes that can cause diseases in the plants. Most of the disease we don't share, the human diseases don't go into the plants, and plant disease causing agents don't usually cause diseases in humans, but there can be some exceptions. Every year, plant diseases cause significant losses to growers. They reduce yield, reduce quality, they increase replanting cost. So a lot of money is spent because of the disease affecting the crops. And like I said, with the humans, us humans too, this COVID-19 issue, when people are more packed, we grow plants together close to each other, we will have higher chances of getting infected. And that happens with for the plants too. When we grow them and they're close to each other, they will get easier sick. The disease spreads easier. And we're going to talk about a little bit of the epidemiology and the plant disease spreading of the disease product as well. Why is disease diagnosis is important? We need to know what's the enemy we are facing. So in order to control that disease, sometimes we have one option, sometimes the other, sometimes we have none. We need to know still that what can we do to make our operation running and without the diseases. And if you're having a disease, then you need to know what to do. Plant disease diagnosis is done by specialists called plant pathologists. There are lots of labs who are running disease diagnostic tests, but diagnosis is more than just the testing. We use laboratory tests to confirm the diagnosis or assist the diagnosis. Joanna also pointed at the field view that she's having when she goes to growers' facility, and she says that don't forget that there is this can be very complicated issues. We may have not only we see that the plant is not growing well. There's some issues with that, but there could be lots of different issues. They could be that primary issues, primary organism that cause the disease, but there could be secondary organisms involved. There could be bacteria or other secondary fungi that are invading the tissues. We may have other organisms present, may have been vectors who actually brought the disease in, insect vectors. We may have other insects present just because the plant provided some started sapping, oozing, so there's attracting other organisms to get into the plant, and you will, as a grower, you just observe those. There's something funny going on. We have a aphid infestation in here, but maybe there's something else growing up. The plant is already growing poorly, is attacked by secondary organisms as well. And then, of course, we have issues of the abiotic conditions that are causing the plant not to do so well. Maybe most common backyard growers issue is watering the plants too much. But I believe that in cannabis industry, there's a lot of very good growers who are taking very good care of the plants and monitoring the growth. So, but it's sometimes very difficult to figure out what is the actual cause of the issue. And that's why we're coming here to help you out with that. That's why you need plant pathology. So what can be the plant pathology and this diagnosis be used for? We often, most of the time, our most sample numbers that we're getting back in here in the lab are, especially from the cannabis samples, they have been coming to solve a production problem. A grower wants to know, or consultant wants to know why the plants are not performing like they should be. Is there something we can do about that? And maybe even like maybe you can avoid the same issue in the future. Then there is a second part of the plant pathology, and what we're doing here is to ensure that the planting stock is healthy. We do a lot of testing for not only for cannabis. This is maybe a cannabis testing part is in the beginning stages for this, that people want to make sure that your source of your planting material is disease-free. But that's why plant disease testing laboratory like us, could be helping you with this to ensure that your planting stock is healthy. And sometimes we react into a real estate purchase deal that the grower wants to know, this is, are the plants healthy? It's the worth of my investment to get that. Maybe I'm just buying something that I don't know. So to make sure that I know everything, that's very common in other industries like grapes, vineyard industry, that people, before they buying the property, they actually do some testing of the plants. And then we do soil testing for pathogens too. It's common use in other industries like the olive growers who olives are really susceptible to verticillium wilt. But here in California, they plant olive trees on soils that previously were grown on tomatoes or cotton, and they want to make sure that the soil doesn't have this disease caused verticillium. The agent verticillium is a fungus, and it can stay in a soil for multitude of years, 10 years. So growers want to be making sure that the soils that they're planting these susceptible trees at, when they put in the investment on the trees and planting, they want to ensure that they don't have this disease in there. So how do we do the disease diagnosis? We are using association with the disease. We're trying to find whether a pathogenic organism is found to be also associated with the plant specimen. So we have a symptomatic plant tissue here, and we are performing lab test. Lab test confirms, yes, if it's a positive, we have a pathogen involved. We have the other option is that there is no pathogenic organism involved. We have a lab test is negative, suggesting that we don't have a pathogen involvement. Then often, I will go and ask questions that maybe there are some other issues that may be underlined that we didn't know. But our tests, at least for the pathogens that we have available, suggesting that there was no pathogen involved. So laboratory tests that we are using here in our lab and are generally used in a plant disease diagnostic. We use microbial culturing. This works very well for the culturable bacteria and fungi, but unfortunately cannot be used for non-culturable bacteria or viruses or viroids. Then we are using PCR, real-time PCR here for detection as well, and immuno-based assays such as ELISA. So how we got involved with the cannabis disease testing was, I think, 2012, when a grower called, Anna-Liisa, can you come and check in my plants? They are just dying one by one. And I think he said that 70% of the plants were showing this type of wilt symptoms, and they were massive, big plants. And I said, sure, I don't know anything about the cannabis diseases, but sure, I'll go and let's go. We'll find out what's going on. And then I used my knife a little bit, and I thought, okay, wilt, maybe there's something in a vascular system. And I peeled off the bark a little bit, and yes, you could see this canker showing on these matured big trees, a little bit of the crown. And further later on, I found out that there was the isolated Fusarium oxysporum from these tissues. So that came my first finding of the what cannabis disease, and it turns out, in my opinion, that is the most common issue with the cannabis bothering and cannabis growing at the moment. There are other disease-causing agents of fungal diseases of the bothering cannabis, such as pythium is quite common, especially in the hydroponic system. Rhizoctonia we found, phomopsis, these are the agents that are causing cankers and the diseases of roots and a crown. And then yeah, maybe very familiar with the diseases of leaves and flowers, that these are some examples. Here is Alternaria. We have a botrytis, a gray mold, and Alternaria is quite common leaf spot disease of cannabis. So a little bit more pictures of the diseases that affecting roots and a crown. Again, Fusarium, wilt and root rot. This particular case here also had Rhizoctonia on it. It may not, there was a mature plant. You could be able to sometimes see the mycelium even growing on the canker, and really massive rot here. We call it canker in the wood. Sometimes it's very subtle. You may only see few roots here. The top part was completely non-symptomatic, no wilt symptoms, anything, but the roots are already starting to turn brown in a tiny potted plant here. And this is a picture of a Fusarium symptoms. Often they start from the roots here and grow up in the vascular system towards the stem. And these are some early symptoms you may see affecting the crown of the plant. More typical diseases, every grower probably battling with powdery mildew. Sometimes there's botrytis issues, also the gray mold affecting the leaves and the flowers of cannabis. Then bacterial diseases as well, quite common. They often cause these leaf lesions. They could be necrotic spots or lesions found, and often surrounding them there is this yellow halo. This is produced because of the toxins that these organisms produce. Common disease-causing agents are Pseudomonas and Xanthomonas bacteria. This is a fairly new bacterium that I wasn't even familiar with. It's called Serratia. I think it was published just past year, maybe even earlier this year, that came out in found from cannabis plants. And then we just published this last year. We found phytoplasma from plants that Joanna was actually brought into our attention. These were grown in Nevada site, and really highly symptomatic plants, stunted growth, modeling, leaf curling, and we found isolated phytoplasma. It's also a bacterium without the cell walls. It's co-systemic inside of the plant. So we published this, our finding, in last year it came out. And later on we also found, and there was a virus here as well in the same plants. And both of these, surprise, surprise, that particular virus, be it curly top virus and the phytoplasma, they are both vectored, spread by insect vectors called leafhoppers. So how do we diagnose for fungal diseases? Sometimes if the organism is sporulating on the plant, so produce the spores, disease can be diagnosed based on the spore structures that are appearing. You may only need sometimes a loop, a magnifying glass, or sometimes stereo microscope is enough to see. These are compound microscope pictures in here, spores of Alternaria, and Orgonium turning into an oospore or Pythium. So they could be seen even without the further lab diagnosis. But what we do in general in a plant, when the plant arrives for disease diagnosis in our laboratory, we take parts, symptomatic parts, we take subsamples aseptically. We first surface sterilize, of course, this specimen, and then taking it with the sterile scalper and tools that we are using in here, we sterilize them first and take some subsamples that we transfer onto growth media that are, we have a special growth media for plant pathogenic fungi, and that allows their growing on the plate, and then also separate media for plant pathogenic bacteria. And then we're letting them grow on that media, and in general lab media for fungi, we can do sometimes just diagnosis based on what grows on the, I'm familiar with the growth pattern. So here is, for example, is a botrytis growing on this media. You can see the gray mold that the sporangia are produced in here, but it also produced this sclerosia here. Bacteria, we are using a specific media. This is a media that allows Xanthomonas bacteria to produce the characteristic colonies. So here is a streak of the bacteria called Xanthomonas in this particular media, and it produced these yellow colonies with this kind of halo around, and that's the Xanthomonas. And of course, sometimes we need to look at this under the microscope, especially to look in the more the sporulation structures of the fungi. So next part, we're going to be talking a little bit about the viruses and viroids. I put together a slide here and in our website too about the plant viruses that are affecting cannabis production based on what is found in the literature publications, that people have actually found the organism and confirmed that this is the virus that is affecting cannabis or hemp production. Also, some of these viruses are affecting also hops. And since hops and hemp are related, it is possible that sometimes some viruses can affect both species. Not all of these viruses we have detected, but not that I'm saying that we haven't done so much of virus testing for that I could have a bigger picture. But some of the virus, like I said, this was the beet curly top virus, was we detected in that Nevada specimen of the cannabis that Joanna brought to our attention. That is leafhopper transmitted virus. It's actually a DNA virus. We have got some specimens that contain this alfalfa mosaic virus in it, and the symptoms here on this leaf that I took as a background here, they are mosaic symptoms caused by alfalfa mosaic virus. Lettuce chlorosis virus is fairly new. Again, this year I found that it is found affecting cannabis. It was detected in a cannabis and actually symptomatic cannabis in Israel, I think. And we do have like sclerosis virus at least here in Southern California, so that's something that maybe we need to pay attention to. Tobacco ring spot virus, tomato ring spot virus, these are viruses that affect lots of crops, especially outdoors. When people are growing outdoors, there is possibility this is in the soils in some locations. Tobacco streak virus is not, we have never detected that from cannabis. Tomato mosaic virus, this is I put here because in the industry there's some talk about hemp mosaic virus. There is no virus described as a hemp mosaic virus, but this is may have been, there's some publications that tobacco virus is a tobacco mosaic virus or tomato mosaic virus could have been causing symptoms on the cannabis. In the beginning, when we started working with cannabis, there were some cases that people are concerned. There were these Agdias test strips that were detecting something in cannabis samples, in symptomatic cannabis samples, and the test strip was showing to be that suggesting that they were positive for this virus, and then we started working with Agdia with this, and their laboratory, they claimed that they found a tomato mosaic virus or related to tomato mosaic virus from that test strip. But I did not hear anything back from that case. I sent some samples for them to do that work, and they claimed that they received that. We were supposed to be publishing that, but I haven't heard anything back from that. So I wasn't, and then next time we buy in a new batch of these test strips, they are not detecting anything in the same samples that we detect something before. So I think that there may have been an issue with the test strips, that maybe it was cross-reacting with something else other than the tomato mosaic virus. So I do not know, because since now that we have the PCR assay, we have not been detecting anything from cannabis. So that may be a virus of a cannabis, maybe not. And then the last here is a hop latent variant, a fairly new finding from a cannabis, but even it says that hop latent variant, it's a variant that it is not showing any symptoms in the hops, but apparently that was a dark art nursery, I believe, in that they published their findings, and they were able to show that there is actually all the plants that they had these symptoms of stunting growth. They came up with the hop latent variant through the high throughput sequencing, I believe, and they found that it is actually causing the disease on the cannabis. So a little bit here, a slide showing about the virus, what viruses look like. They are so small we cannot see them by eye, not even under a microscope. We do need electron microscope for that. And viroids are even smaller than the viruses. So viruses contain either RNA or DNA, that's the nucleic acid here inside of the protein capsules. That is these are the virus particles. This is the alfalfa mosaic virus. It has actually, the genome is packaged into 4 different particles. Here is a CMV. This is tobacco mosaic virus, the long rods. And I wanted to show this picture on this coronavirus times, because when I looked at, this is from the hemp disease and pest of the book. Then I looked at that, oh, there is the coronavirus. So yes, there are viruses that affect the humans, a little bit bigger, these viruses. We have a little bit smaller ones affecting the plants, but they're all kind of shapes they come with. Here's the Ebola virus that this looks like a huge, like a nematode kind of particle. Viroids, on the other hand, like that hop-latent viroid, are even smaller than viruses, and they do not have the protein capsule at all. How they survive, they make these secondary and tertiary structures of that the RNA comes up with, and they are extremely stable. So that's the way that they can stay alive without in the environment, that there are a lot of RNases, enzymes that actually chew the RNA. So that is a stability, that's a potato spindle tube viroid here in this electron microscope picture, very tiny little one here. So unlike the bacteria and fungi, viruses and viruses cannot be cultured. There's nothing growing, there's no media that we can increase them at. We need to use different tests for their detection. Viruses can be only detected by methods that target in the genome, that RNA that it's made of. Viruses instead can be detected by antibodies that target the protein capsules, the surface structures. All that, this is the part of the tobavian mosaic virus genome here. So RNA is packed here inside of this protein, coat protein particles, and antibodies that can be used for targeting those structures. DNA, we will talk about a little bit of methods that can be used for detecting our viroids and virus RNA in a little bit. Here is an example of an immunoassay that can target viruses. You need antibody. Often they are made in animals. They can be also man-made monoclonal antibodies. They are bound on this type of plastic plate in the bottom of this well. We coat this plate with a capture antibody, wash off extras, the unbound antibodies, and then we add in the substrate, the plant extract, which contains, if it contains the virus particles, it will bind, the antibody will detect it, and it will bind, here is the protein particle that the antibody detects. It binds to the antibody, we wash off anything extra, and then we use in a detection antibody that again, it detects, binds to that target protein. And often this detection antibody is linked with a conjugate, that enzyme that when we add in the substrate here for that enzyme, if all this is happening, there is virus present, this sandwich builds up, and we can detect this antibody protein sandwich by adding the substrate, and it will release a color. Often it could be, depending on what kind of substrate we are using, here is a blue, there's yellow, orange kind of colors as present as well. These are also examples of immunoassays. This is Agdia's immunostrip that can be used for quick test of such as TMV, tomato mosaic virus, Phytophthora. I believe that Agdia has many of these, and now they're very handy to have. They are quick, they are field ready. You don't need a specialized lab to do any testing if you're suspecting some. Here is the little bit a GE slide that I got out from the internet, and also in that what kind of the antibody is actually bound here in this, and they used all kind of technologies, and then your sample extracted, this kind of moves here in this it's a lateral flow device from one end to another, it sucks in and it meets the substrate meets here. If there is a virus in there, it meets the detection antibody, and you will get that the signal released in that assay, this line here. This sample here on my picture though didn't have the positive line showing, and only test line showed. So next detection method that we are using very commonly for probably for anything is a PCR, abbreviation for polymerase chain reaction. We actually use here a real-time PCR. It only means that we are detecting, we can actually see, we don't use any gels to get the check for the product. We are using a real-time image of the product developing. So how do we do that? We do need to do RNA extraction. Often these viruses are RNA viruses. They are some DNA viruses, but in general, we need to extract the nucleic acids, RNA and DNA from the plant. And at the same time, we will extract the viruses. The viroids also get extracted. And in order for PCR to happen, we need to make a DNA copy of that RNA. And after that is synthesized, we will start PCR. We use thermostable polymerase enzymes and small sequence-specific oligos. They bind here to the target if it's present, the target virus. And we use heating. The machine heats and cools and heats and cools with the several cycles, 35 to 40 cycles. And during this time, if the template DNA is found, the oligos bind to it, the polymerase makes a copy. Then again, we heat it. We make and let it cool down so that another cycle is made and another copies are made. And that way we can get the exponential amplification of that virus target. And this can be visualized by fluorescent dye that is in that reaction mix. And we can see that developing here. You can see this is a dilution curve of one virus. Here, without dilution, we're detecting here around the 12th cycle of that. After we did the 10-fold dilution, we detect in about 3 cycles later, about 14, 15 cycles. And again, this is 100-fold dilution, 1,000, 10,000. So this way we can see that how we can, it's only the detection is specific, but it's just the delayed when there's a little bit of the virus left. So good thing about the detection of the viruses and viroids, they are actually, when they are present, they can be in the millions. They are fairly easy to detect. Here are some older slide of what are the advantages of that running the PCR test. They are much more sensitive than the immunoassays. We can detect very little bit of the virus. Here, after this cycle already, often the immunoassay, it doesn't respond, there's negative results, but the PCR assay will still suggest that there's viruses present. We need very little piece of plant material to do the testing, and it can be detected year-round, and it's very quick assay to develop, as long as there's a little bit of a sequence available. Like for this coronavirus that is bothering us right now, they developed this assay very quickly. The immunoassay, the PCR assay was developed very quickly for its detection. It can be more costly than ELISA, although I think that those strip tests are also fairly expensive. But because it's expensive, because we need to have all these machines and technology to get the testing done. One downside of with the RNA viruses, especially, the PCR could be, because it's so specific to the nucleic acid, it doesn't really allow too much of a mismatches if the virus mutates at the site where the binding is binding of the oligo, the detection oligo is taking place. So then there could be a chance that the virus is not detected. So that's why we in our lab we're still running that some of the viruses with the ELISA test as well, just to see that if there is a variation that the PCR cannot pick. So, and finally coming to the management part, like I've been talking about all these, that it's important to know what the enemy you are facing. You need to know whether it's a fungal disease, a bacterial disease, a viral disease, and you need to know how that spreads. How does it move? How did I get it here in my production? And in order to, especially in order to figure out if I can reduce the movement or prevent the spread. Here is we come to the epidemiology of the disease. Epidemiology is, here's the definition of it. It refers to how the disease or organism is distributed. I would say how it spreads. This is a disease triangle. That is a very classic plant pathology presentation. In order to disease to take place or occur, we need to have pathogen present, we need to have susceptible host and favorable environment. Without, for example, if we have a pathogen present and a host, but we don't have a favorable environment for the disease to occur, there's no disease. If we have pathogen present and the favorable environment, but we don't have host, favorable, susceptible host, there's no disease. One way of a very efficient way of controlling plant disease is using resistant plants. But I think that this is not, for many of the diseases, we don't have resistant cannabis plants against Fusarium, for example. So, but maybe somebody will do that research in one point and screens enough of strains that could be tested against the Fusarium wilt, and maybe then one day they will be resistant varieties of that cannabis available for growers. So, and of course, without the pathogen, we will have a susceptible host and a favorable environment. But if we don't have pathogen, we have no disease. I wanted to show this with the case of powdery mildew. It's it's so multi-present, it's everywhere. We have that if you are growing a susceptible host like a cannabis, you have a pathogen everywhere. Only way you can actually control this is by changing the environment, making more airflow in your growth. So this can be even managed without adding any fungicides, by just the cultural practices, controlling the environment, making that environment less favorable for the pathogen growth. Management of viral and viroid disease, however, there's really nothing you can do if you get in a virus or viroid into your plant. You cannot do anything about that. If this is that really nice clone that you have, you can have it cleaned by tissue culture laboratories. They can use some practices in there that can eliminate the pathogen, the virus or viroid, from your favorite strain. But there's really nothing that you can do if you get in the virus or viroid into your plant. Therefore, all the management is here. What we can do is to avoid getting the pathogen into your plant material. So that means you should be controlling insect vectors that could be introducing the virus into the plants. And of course, we can try to prevent the spread between the plants. Let's say that we found that one of the plants had a hop-latent viroid in it. We, using sanitary practices, we clean out hands and tools between the handling of different plants, so that we don't transmit the viroids from one plant to another. Management of Fusarium wilt is really nothing you can do about. It's my opinion. Maybe Joanna can share some information on that, what she knows about the managing of Fusarium wilt. But most of the time when you get infected, your plants get infected Fusarium, it's just all you can say that we're trying to elongate the time that the plant can, until the flowering is done, that your crop is harvested. Since there are no resistant or tolerant strains of cannabis available for the growers. So pest management again is to prevent the fungus of entering your production. And how do we prevent that? This organism produces millions of spores. They can be airborne. But I do think that the most of the Fusarium infections, this is no studies done on this, that I do believe that a lot of the young plants are getting already infected in a rooting machine, which provides a 100% humidity conditions, which is basically water. It multiplies very quickly. We had one client do this many years ago, testing the rooting water in a rooting machine, and even after 1 week, the water contains a small amount of Fusarium, and then later on, after 2 weeks in a rooting machine, they were millions of Fusarium spores in that water, and that basically inoculated every single rooted cutting in that machine. So to avoid that, you should not, you try to make sure that you are not adding any Fusarium. Even just one infected cutting can inoculate all the rest of the cuttings in that machine. Then in the spores can be also spread by insects from pot to pot. Insects such as fungus gnats can transmit these spores. And it's quite common with the Fusarium diseases that they get spread, the infections are aided by nematodes. These are little organisms that poke the holes in the roots, and Fusarium is not so aggressive that it can be doing its holes by itself, that in that root zone nematodes can assist. They can poke more holes, and Fusarium that way have a great access to the roots and infect the plant from there on. So,
hey, Anna-Liisa, yes. I have a quick question from one of the attendees about Fusarium wilt, if they've had a problem in the past. Is it still in the soil for the next season's plants to pick up? I think that's a great question, is that can Fusarium be in your soil for next season if you've had an issue in the past?
Yeah, it's a great question. Fusarium can produce these called chlamydospores that are meant for long time survival in the soil. So if you are planning on planting your plants in the same soils, and you had issue with the in a previous season, I would not suggest planting in the same areas where we had it before. So because of the reason that Fusarium can stay in the soil several years.
I've also had some just anecdotal experience with farmers who use a lot of microbiology to battle Fusarium. You know, outcompeting a harmful microbe can often be a way to you know, lower the colony counts in your soil. But I think I concur with Anna-Liisa here. There's not a lot to do. If you have it, it's a bit of a race till the end. And really the best thing you can do if you have an acute problem is managing the vigor of your plant. So, you know, working on giving your plant as much of a fighting fight to have until the end of its life cycle. But yeah, it can be, you know, it can overwinter in the soil. It's kind of everywhere in the environment. So really, a lot of times, you know, sterilizing your environment isn't really a economical or really realistic. So you have to definitely have a specialized strategy for, you know, how to lower your risk if you've had past issues.
Right. Yes, and often growers have been battling with this issue for a long time, and they go through very strenuous cleaning practices at their facility, and they again having the same issues following year.
There's another question about the nematodes and which species of nematodes contribute to Fusarium wilt infection. Can you speak to that at all?
Oh, I should have been doing my nematodes a little bit better. Anything that it goes in the root can poke the holes into the roots. And I'm not so familiar with the cannabis nematodes. I think that Joanna was, you were one time on this talk, but I can't remember the names of those. But anything is feeding on the roots is making a basically opening for the fungi to enter as well.
Right, and we use a lot of beneficial nematodes like Steiner nema to eat up pests and stuff. So those aren't species you need to be concerned about. No. You would mostly need to be concerned with, I think the most common root nematode I see is just the root knot nematode. I think that's one of the most common ones I see with cannabis. So yeah, I'd say that's the species that contributes, I think, mostly to impacting cannabis. So yeah.
Right, yeah, so I'm talking about the presence of plant parasitic nematodes. So those are the ones that are feeding on the roots and poking at the beneficial nematodes, not. They're just there in the soil and making the environment, they're actually eating they're meant to be eating and fighting other organisms in the soil. So those are not the one I'm talking about. I'm talking about plant parasitic nematodes that can, when they're feeding on the roots, they provide entry points to the fungi to get in. Yeah, so best way of prevention is to get a clean plants, and I think that that's often issue that when people have done a lot of cleaning in their facility and they still bring in material that had Fusarium. There's no other way I can think that how can this, but if you have a completely a system that you had the facility that is cleaned completely, and then you're still starting a new, somewhere the fungus came from. It's not, most of these are not airborne infections, unless you have a close by a plants that have Fusarium in it, and you have these insects that can bring it into your soils again. So disease prevention, continuing here, the best way to prevent any of this disease is to start with the clean planting material. You should work with a reputable nursery or source where you're getting your clones from. You should be able to visually inspect that the mother plant is healthy. Ideally, nowadays, you should have some sort of record of testing that no bacterial, fungi, viruses, or virus were detected. We are working with some tissue culture laboratories like Node Labs. Here is a picture of the plants in here. They are doing pretty intensive testing, and I'm sure that they can, and other sources where you're working with, they should be able to give this information to their growers, their clients, that their material, that what they're buying is actually being tested against the viruses, fungi, and so on. And again, you should be inspecting your planting material visually, and you have always option as a grower to run lab tests before you are investing a lot of money onto your plants. Then again, starting with the clean environment, don't reuse your soils. Well, we said that some of these pathogens like Fusarium can stay in a soil long periods of time. Of course, monitoring the growth and the growing environment, especially outdoors, but also indoors. So you need to monitor and control the insects. Remember, they can be bringing in diseases. Then also handling of their plants, you should remember that you can be the one who can be transmitting the disease from one plant to another. So here comes to place that you need to wash either your hands and tools between the handling of different plants, or do use gloves, change them or sterilize them. Most often you can just use wipes, the Clorox Lysol wipes, 70% ethanol, though isopropanol will work as well. You should remember to organize your workflow from the clean areas to the dirty areas if you have a different kind of facilities. You should also practice quarantine. This was actually a great suggestion from Joanna, that this is when you're introducing new plants to your facility, you should first monitor them before growing them together with the rest of your healthy plants, because that's a newcomer. They may have some issues that you did not know about. So you keep them in a separate growth rooms until you're sure that they are healthy, and you can grow them together with your rest of your plants. Then about the disease testing, the sampling is really, really important. This is very last slide of mine here. How do we get a good diagnosis on the plants? Of course, it makes sense that if you see a leaf spots or yellowing on the leaves, you should submit the leaves and stems showing those symptoms. Most commonly, if they are bacteria or fungi that are causing those symptoms, leaf spots, and we can grow them out from those parts of the plant. And therefore, I can say that if we isolated alternaria from that leaf spot, it's alternaria leaf spot. But how to diagnose for the fungal disease that affect the vascular system, such as Fusarium wilt, we need actually samples that consist of the crown, a little bit above the soil line, and then roots. These are often this Fusarium grows up here, but it doesn't grow all the way here on the top. So if you're just taking leaves from here, the symptomatic leaves, even the yellow leaves, they don't provide a good answer for me. They just get in yellow because something is blocking this vascular system, the Fusarium or whatever organisms present in here. So we cannot diagnose properly for that disease without having the parts of the crown and roots in a sample. Viruses and viroids, however, can be detected from the everywhere in the plant, at least from the matured, fully expanded leaves. There is just the way that the plant grows, there's the least amount of virus in the meristemic culture, in the tip. So there's a gradient often happening that's why the younger leaves are not necessarily the best ones. But if you wanted to, and sometimes, like with the phytoplasma, it was very critical. It was showing a critical, very big symptoms, but we did not see that. We examined the plant everywhere, the roots and the stem and lower and part, it was really random, the distribution of that phytoplasma in the plant. So less so much on the viruses and viroids, but it's still, it's possible that some part of the plant just didn't get the virus or viroid in it. So it's good to get few samples throughout the plant, few leaves with the petiole. So we getting a proper diagnosis for that disease.
Yeah, and it's important to here just to interject on that sampling. You know, sampling is your key step in getting an accurate diagnosis. So
Absolutely.
if you aren't sure what your plant is suffering from, getting somebody like Anna-Liisa a sample from all over the plant is really your best bet, because she can take a look at the symptoms and have sample from there. So if you aren't really sure that you're doing a virus screen, or you're not really sure if it's Fusarium, wilt, or etc., the best bet is just to sample all over and really try to get a representation of the whole plant species. So I have a question here. Are there any direct studies of pest and virus effects on yield and quality in cannabis production specifically? Do you have any yield reducing studies that have been done, or if not, can we infer values from similar crops?
I have not followed that if there is any direct studies. I know that there are, like with that hop latent viroid, that was clearly, they had already showed that it is causing stunted growth, and therefore also can be causing the yield reductions. But I think they're just, people are referring to the, I don't think that anybody had done, not to my knowledge. So we are using mainly that with different information from different crops. Like with the Tobacco viruses, they are affecting the flowering. So it is, if you have that virus, therefore it probably has effect on your flowering in this cannabis plants as well.
Okay, great. I have another question on Fusarium. And what are your thoughts on the ability of Fusarium species to become systemic in mother plants and be transferred to clones?
It grows somewhat in a vascular system here, in a stem, and into the lower part. I do not know. I don't think that anybody had done this. So it was surprising to me that there sometimes we can find in a specimen further up here, but often even they have been symptomatic. When we are talking about a big, really big plant, a big tree actually, the cannabis plants that I've seen, that I'm imagining that could be also, the mother can be fairly big and a large plant. It is not growing everywhere in the systemic. It's not necessary that I don't think that it can be found in a very top parts, but I don't know how far it grows. So it grows inside a vascular system of the plant, and it grows into the, for surely in the lower stems, but how far up it grows, I do not know. But like I said, when you're sampling here from the top and branches, even they are showing the yellowing symptoms, we have not been able to isolate the Fusarium from there. We have a better luck of isolating here from the lower part.
And can Fusarium spores be killed by heat, is another question that came through.
If they would be on a surfaces, I think that they are not gonna last too long. Then heat would probably kill them. I mean, we are killing all our cultures in an autoclave, but that's like extreme heat. They are using soil solarization for getting rid of the some soil-borne fungi such as verticillium and Fusarium with other crops. The soil solarization is done with the heating like raised beds. You're heating that soil with the aid of sun, you're heating the soil under the plastic, and it's aiming to kill the, and it does kill the spores. Even these resting spores, those chlamydospores, can be killed. But on the other hand, then how far the solarization goes, how deep it goes, it's not. It may be effective on the surface layer, but no, how is it? Even how many inches down you can get that effect of the solarization, I do not know.
There are some pieces of equipment out there to do sort of like a steam injection treatments for the soil as well, which may aid in getting more depth to a sterilizing kind of action.
Yeah.
But those pieces of equipment are, they're expensive, and I don't know anyone locally who has a steam injection soil sterilizer unit.
Right. Yeah, and then we're talking about that the Fusarium is affecting roots, and we need to treat the soil at least to the root zone of that plant. So that's quite deep with the bigger plants.
A couple people asking about whether or not viruses and viroids or other pathogens, can they be transferred through seed?
Some viruses can be seed transmitted, and now I should have done a little, my homework a little bit better in here, that whether these viruses that affecting cannabis or viroids, can they be seed transmitted. A lot of it's already known from the nature of these viruses, but that they're affecting, how they're affecting other crops. So I believe that some of this information is available already. Hop latent viroid, I don't know whether there's information on that yet. That would be a good way of testing, maybe taking seed from the mother that is a seed producer mother that is having hop latent viroid, and seeing that if it's the new seed that is growing from that, is it free of the hop latent viroid or not? Because these are carried, it's not only that if it's on the surface of it, you know, it could be already inside of the seed itself. So it depends on how they are. They often, many viruses are not seed transmitted, but some are.
So I'm not sure about for cannabis specifically, but I have read some transmissibility for hop latent on hops. And it is transmissible, but on a lower rate. Oh, and here we have a participant saying hop latent viroid is seed transmissible around 10% for hop mothers. Seems to be similar in cannabis. That comes from Jeremy Warren of Dark Heart. Thanks for chiming in, Jeremy. That's awesome. And then again, echoed is about a 10% seed transmission rate in hops for hop latent viroid. So again, that kind of matches with some of the peer-reviewed articles I've read about seed transmissibility in hops being possible, even though it is reduced and it is lower.
Right.
So I have some other questions here. You know, how common is pathogen and viral testing in cannabis production, and how often is there an unknown pathogen discovered? That's a great question.
Right. The most, so I mean, more, like I said, that material that coming for our lab is mainly coming for, there's something wrong with it. There is they're not growing well, they're not looking good, and therefore we're getting a sample and see that to diagnose what is going on. So often, so far, most common we have had, people had issues is with the battling with Fusarium, some fungal diseases. Often they're also bacterial diseases, are common. And the hop-laden viroid seems to be quite widely spread. Viruses, like I said, that I have not seen that much of the, it's only maybe that the alpha mosaic virus, like we had few incidences that we have had the viruses also present, but also people have not done that much of screening of that against the viruses either. And now, how often a new pathogen is found? So far, I think that there is a more research also done on the cannabis. It's every year we have actually new findings coming out that the literature-wise, there are new publications. Even our lab, we have just found last year that phytoplasma, and then this year we have sent our publication in for armillaria and oak root fungus. We found from one specimen that actually Joanna sent to our lab. So we have been involved with already with the publishing 2 new pathogens on the cannabis. With the viruses and viroids, I believe that there is more to be found. And we have the technologies nowadays, like that was used for finding the hop-laden viroid in cannabis. Sequencing, you can just maybe as a growers, it's more costly thing to do, but the finding out the good clones and the starting materials, it could be valuable investment just to screen them against anything in that what's in the plant. And from these kind of searches, new things pop up all the time. So it's new field, a fairly new crop, and people are even it's a very old crop of growing, but the new things are coming up with the current technologies. We find them new findings all the time. So I wonder, did I answer your question?
Yeah, I think so. I have another question here. So many growers reuse their soil. Could a healthy living soil with a diversity of bacteria and fungi outcompete fungal and bacterial diseases, or maybe even virus and viroids?
Yes, I believe that there's always compared to our own gut. If you have a good microbiome here inside, we battle a lot of disease-causing agents. So I think that if that soil is amended with a good amount of nutrients, because the previous crop already took it, you need to add more in. But if the microbes are there, I think that it can be used if there is no pathogens in the soil.
Yeah, and also, having an organically managed, or regeneratively managed, whatever word you want to use, managing a plant to give it a lot of vigor helps quite a bit in just having a strong, vigorous plant helps a lot in fighting most pests and diseases. Now, I have a specific question about powdery mildew. Can I even pronounce that? Sorry, guys. Is the Golovomyces the only PM species we are seeing in cannabis? And is there such a thing as too much air? Airflow. Mm-hmm.
Yeah, I read that there's actually at least 2 forms of the cannabis, 2 species of powdery mildew affecting cannabis. So I'm not sure that I'm not diagnosing them to the species level. I'm not a powdery mildew specialist in here. Is there too much air regarding the powdery mildew management? Probably not. But then if you start having too much air, you reduce in the leaves of the plant. You reduce in the plant photosynthesis activity. So I guess they could come into the limit that you're taking too many leaves off to have the airflow on. So in that point, yes, that's not good direction. But usually it is a disease that you can actually manage with more of the air movement. And you can control the environment.
Yeah, pruning, I think pruning is a big practice that can help combat powdery mildew, but also just the environmental, avoiding temperature extremes. You know, that's a big one for powdery mildew when we get to having, you know, hot days and cold nights and that drastic sort of temperature shift. And, you know, a lot of times if we're in a greenhouse somewhere on the hill, there's not a lot of ways to sort of, you know, even that out or flatten out that extreme change. But if there is a way to control that temperature extreme, that's also a good method in aiding in combating powdery mildew.
Yes.
And I have another question on just microbial competition. Is there anything that eats Fusarium? And what about ideal competitors?
Well, that eats Fusarium I mean, anything that it's if they're probably these nematodes that you mentioned that are good beneficiary nematodes. There's a lot of them that they love eating fungi from the soil and bacteria from the soil. And like with any disease, one spore most of the time cannot do too much. So if you have a competition for a few spores gonna be eaten by something in the soil. We talking about very aggressive strains of Fusarium could be there too, that we have very small numbers per gram of soil, for example. They can cause the disease, but most of the time that we are talking about, it's large numbers need to be present and in good conditions. Like I said, like this rooting machine is for sure a great idea of getting the Fusarium into there's nothing else it can do except water in the roots. And of course it's gonna attack in there. But in the soil, there's a lot of competition going on. So I'm sure that there are organisms like that beneficial nematodes that can be affecting maybe some bacteria too. There's, you know, that's the millions of different kind of bacteria in the natural soils.
Yeah, I've definitely found some success in just using, you know, really high quality compost teas. Certain targeted inoculants are great, like trichoderma, bacillus blends. You know, anything that's going to increase the diversity of your soil is going to increase the diversity of competitors, and that's all going to work to your advantage.
Yes.
So I have another question here about hop latent viroid. Has hop latent viroid been found in Humboldt County or any other viruses of concern? Yes, I'll just answer that. Yes, it has.
I think we got it from some of your samples too, whether they came from there, that county, but yes, I believe so. It doesn't have a county boundaries.
Yeah, I've definitely sampled and diagnosed hop latent in Humboldt County, also Mendocino. And are there any other viruses of concern? I definitely have seen, issues with beet curly top virus, which that one isn't really too well known yet, but I'm glad that we isolated it successfully out of some samples we sent. And, you know, it's not a virus, but it's still a systemic pathology of concern, and that's phytoplasma. And just going back to what Anna-Liisa was saying in her talk about pest management, you know, truly this is where the, you know, integrated management practices we all have on our farms come into play, because, you know, pest management is one of your key control points for the spread of all pathology, you know, Fusarium, all of it, all the fungal, bacterial, and viral diseases.
That's true. That's true.
So I have a question about, could you talk specifically about methodologies for controlling viruses and pathogens with California regulations in mind?
Well, controlling viruses and viroids with the California regulations in mind. Like I said, you know, the best way to control this is to start with the good planting material. You know, that's if they don't know how is it regulated, you know, it's better to start here with the tissue cultured plants. We have several different options of getting this type of plants. Then often these, if they're using a meristemic tissue culture very tip, you can actually get rid of the viruses and viroids with a certain method. So, but they need to use, maybe often is they grown in the heating, they apply heat and the conditions so that viruses don't multiply so quickly. And it is possible to clean clones using this method. But I would, like I said, you need to know where the source you are of the, there's good clean sources of the wood, the clones that you propagate, but then you need to know how the mother has performed and it's been produced and how does it grow. So you don't end up with having issues coming with that vegetative part of the plant that you're planning on cloning. So it's the clone that you're planning on propagating. So lots of this, I understand people share material. Though this is a great clone strain, I like it, you will get it, but word of mouth goes, oh, I want to have that too, and so on. But it's very important that you you have a good relationship with your source plant, a nursery who is producing those plants, so you can rely on that good quality plants are coming from that direction. That, like I said, you know, okay, yes, this clone I heard from yes, it's a great nice strain that I want to be introducing here, but is it worth of that if you don't know the disease status of that new clone? Is it worth of bringing that into your production? How would that be? Maybe I'm not the best person to talk about the California regulations part on this. But
well, you know, one thing I've noticed with the regulated cannabis market in California, I'll interject some ideas here, is, you know, we're centralizing our purchases of genetics for our farms through a lot fewer companies, you know. I don't know if you guys remember, but, you know, 10 years ago, sometimes I'd be getting my clone stock from 3 different sources, you know, because maybe one clone producer couldn't, you know, supply my entire order, right? And we had a lot of clone producers kind of scattered all about. And I think with regulation and compliance and licensing and all that, we've really centralized purchasing our genetics through a lot fewer seed companies and a lot fewer clone nurseries. And so, I mean, I just want to point that out because that, you know, that just movement of regulation lays an industry susceptible to issues that will take hold, you know, when we have fewer selections. And so I think it's all about really cooperatively working together with your nurseries or your seed provider to understand, you know, what their practices are. You know, I can't advocate enough for quarantine practices and holding genetics in quarantine while you have them screened for viruses and screened for bacteria and fungal pathogens, and really taking care of that plant in sort of you know, in a separate quarantine space. We're all quarantine experts now. So, you know, really, you know, that 14-day quarantine time is really important for plants too. It's kind of the same all around, plants, people. But I can't really advocate enough for those, keeping an eye out and spending the money to see if your genetics are clean, and also working with your providers to see, you know, hey, what are your practices? And, you know, people will tell you what their practices are because they're doing good things, right? So I feel like we can help each other.
Exactly.
So I think we're going to move to wrap up. If anyone has any final questions for Anna-Liisa or I guess maybe one final question I had, or one thing I wanted to talk about, was the immunoassays and how you spoke about that being a measurement of certain viral pathogens. And would you say the immunoassay is an indirect measurement and the PCR methods you use are more of a direct measurement?
Well, both of them work. You know, if the good thing about when using the immunoassay is that you can just see the assay, that the results right then. Like if I'm talking about those immunostrips right now, that the viruses often multiply to the millions, and they are easy to be detected. If there is a good antibodies and assay available, immunoassay could be a robust assay for detecting, but it detects the virus, the protein particles, the code protein particles. I would not, the genome, the assays that we're targeting, and the genome is that's the PCR that we are using. It is very direct. That's the genome of the organism that we are detecting, you know. But on the other hand, I think they both are detecting directly the virus. I'm not sure that if it's, I mean, it's just a little bit different parts of the virus that it's detecting. Yeah.
Great. Well, are there any closing remarks that you'd like to give, Anna-Liisa?
Well, I say that thank you for, I want to thank everybody for listening, and thank Joanna for moderating this thing and having me to, and Daniel asking me to come and talk over there for you. It's a great opportunity for us. We have most of the time just dealing in this, we're getting a little bit of sample here and we diagnose. Joanna can see bigger picture. But it's a great way of, I feel that, educating growers on the disease issue, that it is a possibility, it can be a serious thing, but there is also ways that you can avoid disease entering your production, and you hopefully you know what the ways that you can take care of that disease issue or actually prevent it in the future. You have options available for testing to diagnose that yourself, sending it to the lab. I don't mind. Often I getting more information over the phone. People consult before the sending the samples in. What I'm asking, what the kind of symptoms you had it in the past history of the plant. So it's always helpful to contact the lab as well. Maybe you're working with the consultant already. That's fine. But I don't mind actually at all, you know, also consulting over the phone, especially with getting the good sample into laboratory so that we can do a proper diagnosis. And somewhat in the end, when I'm releasing the results, there is also some consultation available in that point. Often, like I said, there's not too much you can do about when you have the disease detected, but we can, I can also talk about the results and what does it mean and how to avoid this in the future. So given that, I think very exciting, this industry is moving on in this direction also, that not only acknowledging that the disease-causing agents are present, but there's also that what we could do about it. It's like I said, know your enemy, and you will go from there easier to you can protect your crop. There are ways to protect your crop. So with that, I think I want to thank you for having me on this seminar to give a talk. Thank you, Joanna. Thank you, Danielle. Thank you, Natalie. And if that's it.
Thank you so much, Anna-Liisa. It's fantastic. I have worked with you for several years, and it's just an absolute pleasure to have you come out on the internet webinar and talk to my community of growers. I absolutely love figuring out and solving some of the mysteries we've solved over the years. I feel like sometimes you and I are plant detectives, which is a great thing. And, you know, thanks everybody for the attendance. The attendance was fantastic, and I hope you guys learned a lot. And if you guys need any support in figuring out how to sample or unraveling a problematic mystery on your farm or garden, you know, you can always contact Dirty Business, or you can contact Anna-Liisa. But, you know, there are resources out there, and the best thing we can do is help to educate each other and collaborate with our observations. So with all that, I'll hand it back to Natalie. Natalie, thanks so much for having us today.
Awesome. Thank you guys. And this has been a pleasure of a conference to produce with a lot of great information. And just want to remind people to make sure you're signed up for tomorrow's webinar on tissue culture and meristematic restoration that Jeremy Warren is going to be presenting on. It's going to be piggybacking a lot of what's already been talked about today and just continuing that education. So make sure that you're registered for that. And with that, we'd like to say thank you to all the attendees and have a great day. Enjoy the sunshine.
All right. Bye. Take care, guys.
Bye. Bye.